Okay, I think everybody managed to get in. Again, sorry it took a little bit longer today. I had some issues with Zoom. It's been a long time since I last hosted, so that's the explanation. So welcome to another edition of Paulo Perks. This week is a pleasure to have with us Gabriel Osés from the University of São Paulo, and he's going to be talking about applying cutting-edge technology to investigate exciting research topics in biomineralization and soft tissue preservation. So moving in, to those that are here for the first time, or those like me haven't been to a PERC seminar for a long time, I'm going to go very quickly through the format of this seminar. So we start with this very short welcome and announcements with a few slides for about 5 minutes. And then we proceed to the most important part, which is Gabriel's talk, which is going to be followed by a more or less 10-minute session of questions and answers. For those that can stay, we're going to have tea time this week. Gabriel agreed to stay with us a little bit to have a informal chat about his research and other stuff, and the talk itself as well. And this is the first reminder that you can send the talks to Gabriel via the chat to our questions at Paulo Perks host, that today is going to be Nina. And so before the talk, a little bit of housekeeping. Paulo Perks values the participation of everyone interested in the paleo sciences. So if you are somehow in this talk and didn't abide by our code of conduct, for example, please go to our website and do that. Again, please remember to stay muted during the talk. You should not be able to unmute yourself, but if you do for some reason, please remain muted. Another reminder that you can send your questions via the chat to our questions host. People can also raise their hands during the Q&A session to ask questions directly to Gabriel. And for doing this, use the raise hands function under I think it's one of those buttons in Zoom controls. I don't remember exactly which. One of those buttons as well allows you to see the captions. It's the CC button. If you press them, they can either show and hide the captions. It's very useful. And we are always looking for new speakers. Please, if you have someone in mind that could be a good speaker for Paulo Perks, please go to our website and check our nominations form. You're also going to see a link to that in the chat in a few seconds. And finally, we also would appreciate very much if you could complete our weekly feedback survey, which is anonymous. It's optional, of course, but we encourage very much because it's important for us. You're also going to be that in the chat in a few seconds. Finally, moving to Gabriel, Gabriel did his bachelor's at the University of São Paulo, bachelor's in geology, followed by a master's in geoscience, same institution, and a PhD in ecology at the Federal University of São Carlos, also in Brazil. After that, he did two postdocs. One right now, it's misspelled postdoc, I'm sorry. The first one at the University of São Paulo, the current one also in the same institution. So with that, I would like to invite Gabriel to take over the screen share and start the talk.
Pedro, can you see my screen?
Yes, we can see it.
Okay.
Have a nice talk. Bye-bye.
Okay, thank you. So first of all, it's a great pleasure to be here today and to be able to talk a little about my research, my current and past research as well. And I'd like to thank the organizing committee for the nomination to give this talk. So today I'm going to talk about applying cutting-edge technology to investigate exciting research topics in biomineralization and soft tissue preservation. The idea is to give some examples of the application of cutting-edge technology to these research areas. And some examples will involve my research, and I'll also give some examples of other research groups that are currently working on these research topics as well. So just to contextualize my research area, it's really interdisciplinary, as you can see here in this diagram. So I work with an area called geobiology, which could be considered as a branch or even better connected to paleontology. And it's right in the intersection of earth sciences and life sciences, particularly geology and paleontology. And basically, I use physics and chemistry principles, specifically technology, to investigate the fossil records. And I've been also working on developing new instrumentation to do that. So as you can see here in this diagram, it's always really important not to focus on one particular issue or particular characteristics of a fossil, for example. Here, the example is of an elephant, of course, but I'm talking about fossils, so we can apply these to fossils as well. So it's always really important to, of course, we can investigate a fossil looking at different parts of it and to its chemistry, to its morphology, etc. But then we have to integrate everything. And by doing this, we can achieve a really or truly interdisciplinary approach. Okay, so that's what I'm going to talk about today. So I'll begin with biomineralization. So we can basically define biomineralization as the ability of organisms to build biomineralized skeletons. That is, skeletons made up by minerals. Okay, so I've concentrated my research in investigating the earliest skeletal animals, or the earliest skeletal metazoans. And there are some questions that we can make to these fossils to understand them better. And these questions actually, some of them are not fully resolved yet. So it's really exciting to study the earliest biomineralizing animals. So we can ask, for example, what was the morphology and composition of these metazoans, the degree of the biological control of biomineralization, and also the biological affinities of these earliest skeletal metazoans, which are from the Ediacaran period, the biological affinities of these groups to Cambrian groups, Sulfanozoic groups in general. Great. So in this phylogenetic tree here, we can see highlighted by this orange rectangle the beginning of this history, the beginning of the evolutionary history of biomineralizing metazoans. And we can see that this history began just at the Ediacaran-Cambrian boundary, and that these earliest taxa were dominated by calcareous specimens, calcareous taxa. So I'm going to show an example of the investigation of the biomineralization of a calcareous taxa from this time interval here, just at the Ediacaran-Cambrian border from Brazil. So we published this paper, I think 1 year or so ago. And as I told you, this paper is about the investigation of biomineralization in a Jackran metazoa called Corbella verneri, which occurs in many continents, many localities, but I've focused my research on Brazilian samples. Great, so here we can see a Corbella specimen. So it's basically a tubular organism made up by sclerites, which are plates and rings, and it had the ability it was highly flexible, and we're going to understand how it was possible in the next slides. So here we have some outcrops from the Tamengo Formation, where these fossils are found in Brazil, in the Corumbá city, in the Pantanal region of Brazil. And the fossils occur in these siliciclastic beds, these beige siliciclastic beds here, which are interbed with carbonates deposited in a marine environment. So here we have the reconstruction of Corumbella tube. And for example, if we make a cross-section like this one in this direction here, we can see the elements which form the skeleton, which are called the sclerites, plates here, for example, in this cross-section here. So we're investigating this cross-section here in the next slides. So and also this cross-section here. Okay. So if we take one of these cross-sections and we make a thin section like this one, we can see some skeletal elements, but of course they are not well discernible from the host rock. But then if we apply this technique here, cathodoluminescence, which is a technique that the application is widespread in mineralogy, sedimentology, we have something like that. So I think you can see that this technique is really powerful to show morphology of this fossil and of other calcareous fossils as well. And now it's possible to see that the wall of the Corumbella's tube is built by individual sclerites, which are not connected together, and they're organized in two layers. So we call these a bi-layered wall. Also, we investigated the composition of Corumbella. So here we can see thin section as well of the skeleton. And we applied micro-Raman spectroscopy using this equipment here, for example, to map the organic composition of the tube. So it's possible to see that the skeleton has kerogen and also carbonate, specifically calcite. And these calcite elements, they are outlined by iron oxyhydroxide films. And when we apply scanning electron microscopy, we can see that these iron-rich films, they're formed by pseudomorphs after thromboidopyrites. And we can see also some diagenetic imprints in these skeletons. For example, here, the arrow indicates a region that was silicified. And very interestingly, by applying EGS coupled with scanning electron microscopy, we were able to note that the most preserved region of the skeleton, which is the inner region along this line here, there's a significant concentration of strontium. So this gives a clue of the original composition of the skeleton, which is one of the questions that we can make to these fossils. We also apply DBSD, which is a technique that is coupled with electron microscopy. And by applying and by analyzing these Cornbella sections, this technique allows us to, for example, characterize really well the boundaries of each calcite crystals that formed the skeleton. And more interestingly, this technique is really useful to understand, for example, crystallographic orientation of crystals that form fossils. So if we map this area here, for example, we have this crystallographic map of the calcite crystals, and we see, we can interpret by the homogeneity of the colors here that the crystals are really well oriented. They have a constant or consistent orientation, as we can see here in this, by the groupation of these colors here, these points, in these polar figures. So we interpret these as being evidence of a biological control in biomineralization, because we know in living taxa that when crystals have a consistent orientation, this is evidence of biological control of biomineralization. And we know that this was the case for the earliest skeletal metazoans. Also, BST enables us to, for example, find evidence of the original mineralogy of skeletons. So we can see here, indicated by the arrows, blue spots, which are aragonite relicts embedded in neomorphic diagenetic larger calcite crystals. So basically, we interpreted that aragonite was the original mineralogy of the skeletons. And then after recrystallization, calcite was formed, these red crystals, large crystals here. But the original aragonite crystals, they were preserved within these calcite crystals. And this is supported by electron probe microanalysis. So, for example, when we consider the strontium of the inner region of the sclerites, the regions with the really well preserved regions, we see really high concentrations of strontium, around 12,000 ppms. And this is really strong evidence of an original aragonite composition, okay? And then to understand better the morphology of the Cornbella skeleton, we applied synchrotron radiation, micro CT. And then by applying this technique, we can see that, for example, along this cross section here, we can see something like that. And then it's possible to note that the wall, as I told you earlier, by using cathodoluminescence, the wall has a double conformation, morphology, sorry. So we call this a bilayered wall. It's also possible to note that the edges of the skeleton, of the sclerites, they're continuous, and there is a gap here along the midline of the tube, okay? So we can see this here as well in this micro-CT reconstruction. And then by analyzing different slices along the tubes, we were able to show that the cross-section of the tube is formed by a bilayered wall with a continuous edge, a corner, and open midlines like this. And as I told you, the skeleton is formed by the sclerites, and in a longitudinal section of the tube or cross-section of the sclerites like this one, like the ones we've analyzed with cathodoluminescence and the other techniques, it was possible to note that they have this morphology. So based on this new reconstruction, we called this morphology or this architecture of the Corumbella skeleton as a cataphractic skeleton, based on the morphology, or not the morphology, but on the protection, the armor of ancient knights. So as you can see here, this armor was formed by a cataphract arrangement by imbricated and flexible plates. Okay. And this, a new proposal, allowed us to change previous interpretations of Corumbella biological affinities. So previously to our work, Corumbella was Corumbella's affinity was interpreted as cnidarian, or even related to cyanotubulite or cyanotubulitids, which occur in the diagram as well. But this morphology is more reminiscent of early Cambrian taxa, which are interpreted as bilaterians, and also other taxa which have yet unknown biological affinities. So this is a reconstruction of Corumbella. And then moving to the second part of my presentation, I'm going to talk a little about investigating the details of fossilization processes, particularly of soft tissue preservation. So this kind of research enables us to gather evidence of geobiological processes in micro nanoscale, and to understand better morphology, particularly microstructures, and also the composition of soft tissues. So I'm going to show you some examples of previous research our group has been doing, and basically related to the amazing paleobiota of the Crato Formation from Brazil, from the early Cretaceous. And as you can see, the preservation is really amazing. So there are examples of 3D preservation, and also the preservation of column of insects particularly, but there are many other groups which occur there, like spiders, and also plants and dinosaurs and fish, for example. And all these fossils, they come from these very fine laminated limestone here, which was deposited at the bottom of a paleo lake at the beginning of the Cretaceous. So, for example, by applying scanning electron microscopy, it's possible to note basically two types of preservation: periodization and keratinization, that is the preservation by the mineral pyrites, and preservation of organic matter by or as keratin. So here we can see some examples of the pyrite rhomboids that were later oxidized, and now we have these pseudomorphs of rhomboids. And the scanning electron microscopy also enables us to recognize, for example, muscle tissues preserved in these fossil insects. And here we have a zoom of these soft tissues. And also it's possible to note that these soft tissues have been preserved as well by right thromboids. So as I said, by applying these techniques, we can understand the micromorphology of tissues, the degree of preservation, and how they were preserved. The Raman analysis was really useful to characterize which iron-rich phase preserved these fossils. So we found out that these mineral phases, basically hematites, but goethite and limonites occur as well. So basically we have iron oxides, hydroxides preserving the fossils after pyrite oxidation. And XRF was useful to show that not only iron is related to the preservation of the fossils, but also other metals like zinc, lead, and copper as well. And this is really interesting. And we also applied a technique called PIXI, and we have this technique and other that I showed you available at the Institute of Physics. And this technique enables us to make an elemental map of a fossil sample, for example. And we have something like that. So here we have the fossil and then the distribution of some elements in the fossil region and in the host matrix as well. And we also did the same kinds of analysis in the synchrotron facilities in Brazil by applying other technique, micro X-ray fluorescence. And we could map, for example, this region here and show the distribution of several elements in this region. We also investigated the preservation of the soft tissues of fish from the crater formation in another paper. And it's possible to see by using SEM that even tendons are preserved connected to bones, and muscle tissue is preserved as well. And these fossils are so well preserved that we can make real histological sections of the muscle tissues, bones, and of skin as well. And by applying SEM, EDS, and synchrotron micro X ray fluorescence, we can show that the preservation is highly complex. Several elements are related to the fossils, and we were able to really understand how they were preserved, the geobiological processes, the mineralogical processes related to the preservation of these fossils. So now, just going to the end of the presentation, I'm going to show you some examples of the application of different techniques that our group has been using as well. But these results are not related to our research, but they are examples of research of other groups. So other technique that can be used to understand, for example, the morphology of really small structures in fossils is called FIG, which is focused ion beam, and it's basically an ion beam integrated with a scanning electron microscope that can mill or cut really small structures like these microfossils here. And you can do these sequentially and then put all the images together and then have a 3D reconstruction, like a micro CT reconstruction of the structure that was sliced. And if you integrate this slicing with EDS maps of each slice, you can put these maps together and yield the same, but then you have the chemical composition of this structure. So these FIB slices coupled with transmission electron microscopy and EDS show these what I've just said. Okay, so the distribution of elements forming a map in the region that is being analyzed by FIB and EDS together. Another very powerful tool for investigating particularly fossil preservation is coupling X-ray fluorescence and X-ray diffraction. So, for example, in synchrotron facilities, you can merge these two techniques together and have an idea of the distribution of elements, but also, and really interestingly, of minerals related to these elements. So you can have a very good idea of the distribution of the elemental composition and of the mineralogical composition as well. Another really useful and interesting techniques usually related to synchrotron facilities is called XANIS, this technique here. And you can make, for example, point analysis, which we have been doing, but also maps of an entire fossil. And you have spectra of these analyzed points. And if you compare what you are looking for example, if you compare the hairs of these mammal here with some standards of what you are looking for example, melanin, you can compare, generally speaking, the shape of the analyzed spectra with the spec of these of the spectra of these standards, and then you have an idea of the composition of the structure you are investigating. So you can couple these with X-ray fluorescence and with X-ray diffraction, and you have really powerful tools to characterize samples and understand fossilization processes and paleobiology as well. And another technique which has been being employed in the last years, it's a really useful and powerful technique, is TOF sims. So using TOF sims, you can, for example, map crystals, pyrite crystals, for example, or microfossils, and then understand the isotopic or isotopic composition of the minerals and understand, for example, analyzing the fractionation pattern of these compounds. You can have an idea of the participation of microbes in the precipitation of some mineral, for example, pyrites. So you can have an idea of the geobiological process involved in the precipitation of these minerals. And you can analyze, for example, if there is organic matter and the characteristics of organic matter as well. And a really useful technique that has been applied to paleontology in general is CT scan. So you can have a CT scan like this one that we have at the University of São Paulo in the engineering school, or you can have a micro CT or nano CT beamline in a synchrotron as well. And a really interesting example of the application of micro-CT to paleobiology is the investigation of the late Ediacaran tubular taxa, the earliest skeletal metazoans, which appeared in the Ediacaran period, as I told you, as I showed you at the beginning of the presentation. So these two groups, they applied micro-CT to investigate, in this case, Claudina and here Namacalatus, which are two taxa from the Ediacaran. And they could reconstruct the digestive tracts, guts, within the fossils by using micro-CT. And these enabled the researchers to change the biological interpretation of the interpretation of the biological affinity of these groups related to Cambrian groups. So basically, this evidence here supports a bilateral affinity for these groups. So you can use several techniques to understand paleobiology evolution, not only geobiology, of course. And I'd like to show you, just finishing the presentation, some equipment that are available at our lab at the Institute of Physics at the University of São Paulo, both at the LACAPC lab, which is the lab I belong to, and also to the LAMFI lab, the particle accelerator. So, for example, we have a portable Raman equipment, portable XRF equipment. We have a mapping, an XRF mapping as well, which is under upgrade. So it's being upgraded to also to analyze minerals, for example, by XRG. So we're going to couple XRF and XRG together. We have an FTIR equipment as well. Digital radiography, which is really useful when we, for example, are selecting samples to send them to a micro-CT in a synchrotron or in another lab, for example. And we have also a really good and complete updated photography system, even including UV light photography. And we have Pixie at the LAMF lab, which is coordinated by Professor Thiago Fiorini. And as I told you, it's possible to map the composition, the elemental composition of samples using Pixie, using this technique at the accelerator. And the LACAPC lab is headed by Professor Marcia Rizuto. So I'd like to thank you all, and also thank the institutions and people who have been working with me in the last years, who are currently working with me. And that's it. Thank you very much. And if you have any questions, please make questions. I'm going to answer them, and you can check my CV and other stuff in these links here. Thank you.
That's great. Thank you. Thank you so much, Gabriel. It was an amazing talk. I was very, very surprised to see some of those images, the way you analyze the fossils. You can keep the slide, yeah, right there, so you can move to some of these if necessary. So just a reminder to everyone that if you want to ask Gabriel a question, you can either raise your hand or drop a question in the chat to our questions host. We do already have a few questions here. I'm going to start reading them. And Gabriel, just to let you know as well, I'm going to copy and paste those questions in the chat so we can also read them if you want.
Okay.
So I'm going to start this one. Just find the chat. Okay.
So I'll just open the chat to
be able
to see them here.
Okay. So the question is, do you already find biogenic apatite or silica in the diachront, or do you know when this adaptation arose?
Okay, so I've never found neither apatite or silica in the diachront fossils, only silica related to the genetic processes. I think I showed one example in that SEM image. And apatite and silica, I think they became more common as biominerals later, not in the diachront. Okay.
Okay, makes sense. We have another question that came in the chat right now. So could bilateral affinity for Claudina or Corumbella push the origin of metazoans further back into the early Ediacaran?
Yeah, that's a good question. I'm not sure if it would put the origin of metazoans further back, because we in the early diagram, because we already know that metazoans appeared in the diagram, but I think it can push the origin of bilaterians and back to the diagram, and particularly of some bilateral groups, and it's really important. So, for example, Namacalatus specifically was the biological affinity was attributed to Lophotrochozoan. So we have some idea of the bilateral biological affinity of some groups. Okay, they were probably they were bilaterans, and also we know which group they could be related, and those techniques they I think they can help in in this. I mean, it can help understand better the morphology and consequently the phylogeny of the groups.
Okay, thanks. Let's move to another one. So it's
Okay.
also kind of related.
Mm.
So kind of a broad question. Is there a particular fossil material that you would like to analyze using any of these methods?
Mm, that's a really good question. Yeah, many. uh, Maybe understand better the preservation of fossils from the Burgess Shale, and of other Ediacaran biomineralizers as well, to compare with Corumbella. I
Berkshire
think,
would be nice. Yeah.
yeah.
Uh, okay, let's see another one. This one. So great talk. That Xenis synchrotron map looked really interesting. Do you have an idea about the scanning and processing time for such a big sample with this method?
Yeah, that's a really good question. I think I said I've never had the chance to analyze fossils using Xenis. I mean, with maps. I've never done maps with Xenis because I think it wasn't possible in the previous Brazilian synchrotron facilities. Now I think it's possible, but I've never tried. But the point analysis using this technique, which what I did, yeah, it's not a quick thing to analyze a point in a fossil, for example, using XRF in this using synchrotron. It's much quicker than analyzing a sample, a point, for example, using this technique. So it requires more time, and you scan different parts of the spectrum for each point, so it takes longer than XRF analysis.
Okay. Um, Another one. And I guess it's the last one you have available here. So if anyone wants to make another question, just make sure to
Okay.
send it to the questions host. So the last one I have here so far is this one. Did Corumbella live in an Aragonite Sea? Do you think this was a major control on the type of calcium carbonate that precipitated?
Yes to both questions. I fully agree with that. We have independent evidence that the sea was saturated in aragonite, and I think that this was a major control type of carbonate that precipitates. Yes, I think so. Mhm. And we have evidence not only for the 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 Corumbá region, but also we have evidence for other localities in the world for the same time periods, of course. Mhm.
Interesting.
Okay.
So yeah, I don't think you have any other questions. So maybe we can move forward to finish the talk. So
Okay.
let me just take over the screen share from you.
Okay.
All right. So thank you again, Gabriel. It was great talk. Really enjoyed it. It was I learned a lot, so thank you.
Great.
So I would like to invite everyone that can attend the next seminar. We are weekly seminar series, so we have talk usually every week. Sometimes we have to have a hiatus, but usually every week at the same time. Next one is next week, next weekend Tuesday again. So March 15th, we're gonna have Daniela Dmitrievich talking about reef community responses to global environmental perturbations. Just a reminder that today Gabriel agreed to stay with us for tea time, which is an informal conversation with the speaker. And as a nice breaker question, we're going to be answering which is our favorite Precambrian fossil. So with that, we have a short break of 2 minutes right now, and people can use this to stretch their legs, get something to drink, and I hope to see some of you at least in a few minutes. See you soon. Bye-bye.
Thank you. See you.